Welcome everyone today. It's the April 18th meeting of the IMAG MSM Working Group on Multiscale Modeling and Viral Pandemics in GLIMPRINT. Appreciate you coming this afternoon. I know it's the end of the semester, so people are hectic sometimes. We have one talk this afternoon from Jim Sluka. Looking forward to that. As always, Tom, myself, Jim, and Lorenzo are available to take suggestions, comments, improvements. Please let us know what we can do better. We have a variety of communication channels. They work better if we use them. There is the glimprint.org website that's getting a revamp. There's the Slack channel, which I don't think is that active at the moment. There is a Twitter handle, MSM Viral, IMAG MSM Wiki page, which is our main means of communication at the moment. There's a LinkedIn page also, which people are free to use. And there's our archive of seminars, which has more than 150 seminars at this point. We're working on building up the GLIMPRINT organization and website. If you'd like to help us out, Jim Sluka, our speaker today, is leading a lot of that effort, and I'm sure he would appreciate your help. Again, we have that Twitter X handle, MSM Viral. If you put that hashtag on projects that are relevant, that would be great. We want to announce there is the Foundations for Digital Twins as Catalyzers of Biomedical Technological Innovation. That's due the 21st of June, but there's going to be another round next year. That could be of interest to many people. And there are a variety of NSF calls out now also beyond that one that are relevant to digital twins. There's a mathematics of digital twins as well. Reinhardt is doing a faculty search at the moment in computational systems medicine. That's a great opportunity. People might want to contact Reinhardt if you're interested in that position. We're formally announcing our CompuCell 3D free annual virtual workshop. It's going to run between the 28th and 3rd of August, and then there's going to be a hackathon on the following weekend. We'd love to have people help us get a good turnout. We've had people from all over the world in the past few years participate virtually. Typically get about 60 people joining and about 35 for the hackathon. We'd love to have your students, your postdocs join us for that. There are these two publications of interest. Of course, the National Academy's Foundations for Digital Twins paper finally came out in its full form. That's quite interesting. It's also quite interesting that the Chinese have identified medical digital twins as one of their core technology development areas for the next few years. It's quite interesting to read about that. If you have a publication related to the group and you'd like to share it, please do send it to us. It could be posted on the IMAG site and also the GLIMPRINT site. We want to make sure that you have the visibility that you deserve for participating in the group. Jim Sluka is the one to contact on that. We'll have a steering committee meeting tomorrow. I hope that various working groups that have been working on aspects of GLIMPRINT will be reporting tomorrow. We can work on those together. So we have Randy Highland speaking about the physicell modeling environment next 2 weeks, next time. We're looking for suggestions. We don't always meet in the dog days of the summer, but we will meet going forward, at least for the next few weeks. Please, as always, suggest yourself, suggest your students, your postdocs. Even more, please send us the list of people that you would like to hear from. Lorenzo is extremely good at getting people on the schedule. So if there's a recent paper, either by experimentalist, modeler, data scientist, computer engineer, that you think is interesting, please send us that suggestion and we will do our best to get them on the roster. We really want the talks that we present here to be ones that are of interest to the community, and that works best when you're the ones who suggest the speakers. So This week we have Jim Sluka, Predicting Disease with Broken Models, a work in progress. And I won't take any more time, and I'll turn the screen share over to you.
So I'm going to talk a little bit about a work in progress, dealing with ways of analyzing model output, particular models that generate images as part of their output stream. That's just my abstract, the one that was up on the site. But first, to start off with a little motivation. So, as I said, spatial computational models often produce images as outputs. In general, modelers try to develop a model to reproduce some observation, often a normal state and or a disease state. When the primary output of the model is an image, understanding the range of outputs that the model can produce is often challenging because you end up having to look at a large number of images. If you can interpret your output as some numerical data, some metric that reduces an image down into 3 or 4 or 5 numbers, it's much more straightforward to try to analyze the data. But often models can generate novel outputs as a function of the parameters, and usually the modeler is not interested in those things that are novel. They're interested in reproducing their normal and their disease state. But if you have a model that actually encompasses sufficient amount of biology, it might also be able to reproduce states that you hadn't even thought about to begin with. A little bit of background of where I got started on this. About 2 years ago, I started a project with Toru Matsuura in Japan. He came to me with a spatial model, wanting to develop a spatial model for lung adenocarcinoma subtypes. And he gave me this image to start with. Now, what we have here is across the top is a set of just an epithelial layer with stromal cells below it. There's some abnormal proliferation typical of cancer progression, and it generates different types of cancer topologies. You have papillary, where the epithelial layer bulges out and the stromal cells fill it in, micropapillary, mucus-filled acinar, solid tumors, and lipidic, where just the normal epithelium gets replaced with cancer cells. So he wanted to develop a model that could reproduce all of these various states. And that's what got me thinking that normally somebody would come in and they would want to do one of these models. They wanted a stable epithelial layer that, under some set of parameters, generates some abnormal state. But if your model's good enough, is it possible that you could predict these other abnormal states without actually designing the model specifically to do that? Now, we're not going to go as far as this today because this is a much bigger project. So we're going to do something much simpler. And to recognize in general, the range of images produced by a spatial model is some function of the parameters of the model, as well as whatever processes are included in the model. But the parameters of the model may encompass a very large space and may result in a very large number of outputs. In addition, most spatial models are probabilistic. So even with a given set of parameters, you might generate more than one kind of output. So with the underlying question of how does the modeler explore and interpret a situation where their simulation produces a large set of images, basically, from their simulation. So there's a couple ways you might go about doing that. The modeler might just run a lot of simulations and then manually look at the images. If you have thousands of simulations, that's very time consuming. And it's also biased by what the modeler is expecting to find. They're looking for particular things. Alternatively, the modeler could define some metrics that reduces an image down into some small set of numbers. And then they might do analysis on those metrics instead of the images themselves. Finally, what we're going to talk mostly about today is using a naive image clustering technique to try to cluster the images themselves and not rely on some user-defined set of metrics describing the images. I should say that there are a lot of techniques out there for image clustering. There's techniques in the wild for dogs and cats images and things like that. There are techniques for clustering histology slides, for example, which look at small windows of the image and look for horizontal lines and vertical lines and generates counts of how often those kinds of things occur. But in our situation, our images are a little bit different. They're not as complex as real world images like a dog or a cat. They're not as complex as a histology slide. They're often much simpler 2D images like this one shown here. What we're going to do is we're going to use one of the simplest spatial models for cells, and that's cell sorting is kind of a test case. So this model begins with a blob of cells. We have two kinds of cells, reds and greens and blues. And based on adhesion and a random walk, the goal is to sort the cells into regions. And in particular, to sort the cells such that the blue cells end up as a core and the green cells on top of it. And this is a Computational simulation that we're going to be using. And you see it very quickly sort of sorts. And then after a very long period of time, we'll skip forward in history, it settles down into a properly sorted cell. So we have blue cells on the inside, a layer of green cells on the outside. A very simple, very straightforward. This is one of the training modules that is used in Computational. So we're going to use this as our model. The parameters we're going to explore in the model are the adhesion energies between the three cell types, the red cells, the blue cells, the green cells, the blue cells, and the black medium cells. We're going to scan over 3 values for each of these 5 parameters. We're going to do 3 replicates for every set of parameters. We're going to run Computational out to 100,000 Monte Carlo steps, which is really time consuming. And if you might have noticed in the animation that the sort actually gets sort of done quite quickly. To actually get to the final blob of cells takes quite a while. And then we'll just use the images like this sample here as the output of the model. Given this set of parameters, there's 729 simulations set. As I said earlier, the user might decide to define some metrics to describe the output. And so we're going to define 5 metrics, and they are the condensing cell contact areas, blue contact, green contact, blue-green contact, and then both blue and green contact with the extracellular space. So instead of getting an image like this, which is 300 by 300 by 3 pixels or integers, every simulation will just describe by 5 numbers. So that's the kind of how you would do it if you're going to use user-defined metrics. So go ahead and we crank up the little cluster and we let it chew on that. We generate 722 simulations. 5 of them failed. They crashed for numeric errors. And I just ignored the fact that 5 of them crashed. But here's a random selection of 50 screenshots out of the 722 simulations. You see there's all kinds of different results. There are certain things that seem like they occur frequently, like this one here. It seems like it occurs again and again and again and again and again. And then you get dispersions and various other shapes. So that's our input data set. This is our output of sweeping parameters from a spatial model. And so our overall workflow is going to be to generate some set of images. Start with a model, generate a set of images. I have thousands here. In this case, it's 700 and some. And then we can either take those images and use the user-defined metrics to try to cluster the results, or we'll use unsupervised machine learning to try to cluster the results. And the ultimate goal, of course, being, is there a success image in the sets? Is there a cluster of success images where the simulation did what you want? And in addition, are there other clusters that give suggestive results of ways that the biological system, not just the model, but the way the biological system represents, might fail? As I mentioned earlier, there are other ways of doing image clustering. We're doing this route mostly because I wanted to have better control of what was going on. And some of these other image clusters are somewhat black box approaches. So we have a set of images. The first thing we'll do is clustering using user-defined metrics. So again, for every simulation we ran, we have the input parameters, which we won't use in the clustering, but we have just this set of 5 metrics for how well the cluster went. I use JUMP to do the clustering because it's got a really slick interface. And if you cluster to do principal component analysis, first of all, on these length metrics, find the first 3 principal components cover 96% of the data, the dispersion in the data. And this is how it remaps in the principal component space for the first three components. And you see it's definitely separating into unique clusters. And that could be very useful for an analysis of the results and picking out clusters from here. You can also in JMP do the k-means clustering on those 5 length metrics, and then overlay the k-means clustering onto the principal component analysis. So that's what this is. This is going to told it to do 32 groups. So it identified 32 groups. This very large blob is basically one simulation, and the large region is because with just one simulation, you really can't calculate a 95% probability surface. So it ends up being quite large. But see, you do get clustering and large number of clusters and reasonably well separated. So now we'll do the other approach. We'll try to cluster the image using unsupervised learning and transfer learning, essentially. We're going to start with one of a couple of different possible pre-trained convolutional neural networks. This one's called VGG16. VG16 has this rather complex convolutional neural network layout that starts with a 224 by 224 by 3 color image, and through various layers of convolution, ultimately maps that image into a vector of 1,000 length that is descriptions of the image. So it knows dog, cat, tree, car, etc. So you put a cat picture here, it's supposed to turn on the output node here that says that's a cat. VGG16 was pre-trained with more than 1 million images and maps to an output vector of 1,000 different things that could be in the image, or that the image could represent. So that's the basic structure, but we're going to use it a little bit differently. Instead, what we are going to do is, first of all, we're going to throw away the output vector completely because we don't have images of dogs and cats. We're going to change our input image to be our screenshots from the Computational simulation. And we're going to instead just capture the penultimate layer of the network, which is a vector of 4,096 nodes. So that represents a recoding of this original image. And we go from 150,000 data points, integers, 8-bit integers, in the input image down to a vector of floating point numbers that's 4,096 long. So about a 40-to-1 data reduction. Note that we're not training the network, the VGG16. We're simply putting an image at the input of a pre-trained network and taking the penultimate layer of nodes as its output. So the process is really quite fast. It takes about 1/3 of a second to process one image on my 10-core laptop. But compared to training, which would take days, if not months, this is a relatively fast process. It's all done in a Jupyter notebook. It uses the Keras package, which includes TensorFlow and has the VG16 network as well as the pre-trained network weights. The sklearn package for k-means and PCA, and GraphViz, because every 10 years I have to go back and relearn how to use GraphViz. GraphViz was written in 1982 or something by AT&T Labs, but we're going to use it. So our original images were all 300 squared pixels, which is just how I had the Computational set up to output the images. The notebook ultimately will rescale it down to 224 because the VGG is specifically designed to handle images that size. That's a complication that is always hard to understand if it's going to hurt you or not. You don't really quite know what rescaling an image is going to do to the characteristics of the image. So our overall workflow is we've got some set of images. We pass it into the pre-trained VGG16 and convert every image into a vector of 4,096 values. Take those 4,096 values to do principal component analysis and reduce it down to 100 values. And then use k-means clustering to generate image clusters. So the whole process is similar to using the user-defined metrics, the edge metrics, except our initial set of vector values is coming from the VG16. I've also looked at just going directly and sticking the entire 4,096 and doing k-means on it. Turns out that works about as well as going through principal components and the k-means actually does principal components on the data anyway. It's just a little faster and more efficient to reduce your data down to a smaller set of values than 4096. I had said that 96+% of the data in the user-defined metrics was explained with the first three principal components. With the AI data, with the 4096 vector from VG16, it takes a lot more components to explain the variance. So here you can see, to get to up around 90-ish% of the variance, you need more than 50 components in your PCA versus 3 in the user-defined metrics. So the data is not quite as information rich. The image itself is not as information rich, which is not unexpected. We can then look at the size of the clusters as a function of the number of clusters. So that's the horizontal axis, is how many means clusters we're trying to generate. Starting with one cluster where the clusters are really quite large. And as you make more and more clusters, the individual clusters get smaller and smaller. The red line is just the derivative of the black line. Normally, you might pick a number around 10 as the number of clusters. That's kind of where the elbow is, the point of diminishing return. But I find that I like it a little bit better if I use a lot more clusters than you think you might need. The worst case is you get some clusters that probably should belong together. The positive case is that you make sure that you find very small clusters. If you tell it it's only going to do 10 clusters, it's easy to get a very small cluster sucked into a larger cluster. So that'll be our approach. We'll pick 32 clusters, and that also matches the number of clusters we look for when we're using the user-defined metrics. So after clustering, this is just the size of each cluster. There's a cluster, the smallest cluster, with 3 members. The biggest cluster has 63 members, quite a few clusters in the range of 20 to 40. I remember our total data set is 720 or so images. So this one cluster is almost 10% of the data. And if you go through the first 5 or 6 clusters, you get out through a significant fraction of all the data in the set. This is just the caption for the next figure. We now have 32 clusters. We can look at the biggest cluster. These 5 images all came from the biggest cluster. This is the image closest to the image to the cluster centroid. We call that the representative image for the cluster. Then we have the 3 next nearest to that cluster centroid. And then we have the 2 images that are the farthest from this cluster centroid. So these 2 images are in this cluster, but they're quite farther away from the center of the cluster. So you get some idea of, these are all in the same cluster, and it's still taking these into account out here. These are the 3 biggest clusters. So you see they all look very similar. And together, that's 145 of the total 720 plus images. And they look relatively similar to each other. We can then look at one take home from this is the 3 biggest clusters are all very similar, and they're all random mixtures of cells. Basically failures of the goal of making a cell sorting. This shows all of the centroid images for all 32 clusters sorted in order of largest cluster size. So this is, again, the biggest cluster, cluster 6. The first 3 all look very similar, like as on the previous slide. And then as you go down through smaller and smaller clusters, you get all the way down here to the last cluster. There's only 3 images in this cluster, which happens to be the 3 replicates for one set of parameters in the model. So you see we get a nice range of a nice range of image outputs. Though you might make the point that, well, it sure looks like you got too many clusters because this should probably be the same cluster. But again, we just wanted more clusters than we thought we might need. Once you have cluster centroids, you can generate a distance matrix, so how far every cluster centroid is from every other cluster centroid. And you obviously don't want to look at a table like this. So instead, this is where GraphViz comes in. So GraphViz lays out networks, connected networks. Here, this is the minimal spanning tree. So this is the minimal number of edges that will connect all the clusters together. It starts off with just being the strongest connection for each node. and then augment it to make sure you don't end up with unconnected subgraphs. I've rotated the image so it fits better. That's why the numbers go sideways. So have a nice sorting here, and not only the sorting of the images, but some connection about what the neural net thought made the images different, the image clusters different. You can look at them and kind of make some calls about some further subgroupings, presumably subgroupings that would have been obtained if I had done less than 32 clusters. But looking at these images, you can start making descriptions about what they are. Here in the middle are our two proper sortings, one with the blue inside, one with the green cells inside. So the two ways of doing it, depending on the parameters. But then you get a range of other results. The most common one is the random mix, all the various different ways of getting random mix. Some of the difference between these out here are just what color the cell is on the periphery. Sometimes it's mostly blue on the periphery, sometimes it's mostly green on the periphery. In addition, we have things that I call pandas because they look like pandas, big blobs, but not one cluster surrounded by another, one blob surrounded by a bigger layer. And we get sock dolls because they look like sock dolls. They're not quite as adherent to each other. We get things that look like partial sortings, and usually very similar. They're mapped very similarly by the VGG16. These two differ by blue on the inside versus green on the inside. The cluster is separated, but they're in the nearest neighbor map. They're next to each other. And then we get a group of results where either green or either blue or green forms a blob and the other color is separated. And then finally, the fully dispersed model out here at the end. So we do get sets of understandable things. We get the thing that we expect to see. We get homogeneous mixtures where there's no cell sorting. We get full dispersion of one or both of the cell types. And then we also get these things that are somewhat unexpected, the pandas and the sock dolls, where they haven't quite started to break up, but they also haven't really sorted into the proper sorting for cell sorting. This is the minimal spanning tree. You can also use GraphViz to generate the two nearest neighbors. So here, every cluster gets to connect twice. And that starts to connect up some of the clusters in slightly different ways. Again, we get semi-sorting results here, semi-sorting results here, random mixtures here, coupled through a random mixture. The fully dispersed, blue blob, green dispersed, green blob, blue dispersed, which is interesting that at length these are considered to be different by VGG, and they're considered to be different than the fully dispersed in a way that's obscure. So now we've done clustering two ways. We've done it with the user-defined metrics, which is edge homologous and heterologous edge lengths, and we've done it with the unsupervised AI ML. So now we can try to compare the results with each other to see if they're giving us different interpretations of the image data. This is the same principal component analysis that I showed earlier with the edge length. So the user-defined metrics, just 5 metrics per image, principal component analysis, and then overlaid on that analysis are the VG16 k-means clusters. So that's what the coloring is. And you see kind of at first glance, they seem to have a fair amount of agreement with each other. However, if you look closely into a cluster, you'll see different random colored dots mixed in. Nice, here we have 2 sets of colors very close together. So they're doing slightly different clustering. This ugly graph, this is an attempt to compare if particular images in one cluster and one method is in the same cluster in the other method. Here we have the 32 clusters from the Computational, the edge lengths determined in Computational. So user-defined metrics. The width of the bar is proportional to how many simulation results fell in that cluster. Over here is the color coding for the VG16 clusters. Again, the length of the bar here is proportional to the size of that cluster in VG16. If you look at a particular column here, this cluster 23 in the length user metrics partitions across all of these machine learning clusters in this way. So this cluster is spread out among the clusters in the other clustering technique. So this looks fairly discordant. You find that the very small clusters tend to be done well. So the fully dispersed cluster with 4 members is one of these ones. It's a homogeneous bar. Those are 2 techniques agreed. But I think some of the discordance here is just from an artifact of using too many clusters. That how you break up a supercluster into subclusters is very dependent on the technique that you use. So as it says here, some of the discordance may just be that the cluster sizes are so large. This is another comparison. This is this particular fully dispersed result. This is a bar graph of the 32 clusters from the k-means using the VG16 versus the cluster using the user-supplied metrics. Again, 32 clusters, the length of the bar is proportional to how many images are in that cluster. And if you click on a bar, you get that the just fully dispersed in the VG16 matches the fully dispersed using user-defined metrics. If instead you look at this cluster, this image is in this cluster in the machine learning, the VG16, and this is how it distributes across the clusters in the length-based clusters. Yeah, this is the biggest group here, and then partitioned into the 4 biggest groups in the other clustering, the length clustering. So again, it's probably a result of forcing it to do more clusters than are really probably justified. But that's okay. It's easier to look. It's relatively easy to look at 32 clusters, and you're a little bit more certain that you didn't miss a rare cluster like this 4-member cluster. So as I said, this is a work in progress, and the starting goal was, or the kind of the starting concept was this image here. Where I'm at now is we are going to try to randomly sample parameters in the Computational model. In the paper that we published, these are the these are the parameters within the model that we changed. And then this axis down here, these are the user-defined metrics about does the epithelium cell interact with only cancer cells, the stromal tissue, contact mucus. There's various other user-defined metrics of looking at these images. Again, the user-defined metrics are always biased by what you expect to see and what you want to see. So we want to redo this using this VG16 approach. We haven't gotten that far yet. This is a much more complex model. It has a lot more parameters in it, and it also has a much more complex output image. This figure is from our paper that was accepted recently and shows simulations that start to look like some of these observable tumor topologies. This mucus-filled one, for instance, is this model output. So that's where this work in progress is now. Still talking with Dr. Matsuura to decide which parameters in the model we're going to actually change. We're going to stay with the user-defined metrics because that's relatively straightforward and it was done by an expert, and that's what you want. And once we get that done, we'll go ahead and run the thousands of simulations and try to apply it to try to run it through this clustering technique to see if one, if we can find the states that we expect to find, 2, to see if we can find unexpected states, and 3, to see if there's any mapping between parameters and the likelihood of getting a state. You might have remembered that back here that this particular output was very difficult to produce. It only occurred with one parameter set. Many parameters give this kind of output. This is very common, especially if you start super clustering the clusters. So it's easy to get this, it's hard to get this, and it's an intermediate difficulty to get the proper sorting result. So I think I'm about on time. Conclusion. So compared clustering using unsupervised AI learning versus using user-defined metrics. The unsupervised learning is not constrained by expectations. In contrast to user-defined metrics, where the modeler is going to have whatever their biases applied to their metrics. The AI is somewhat slower, but not terribly slower, since we're not actually training a neural network. And indeed, doing the AI re-imaging or AI remapping of the images is fast compared to how long it takes to actually generate one full simulation run, which runs this particular simulation on a single core takes about 10 minutes. The AI reinterpretation of the image takes about a third of a second. There are a lot of other approaches to analyzing images. They're usually designed to work on real-world images like dogs and cats. There are a lot of tools available for clustering images like this histology slide here. Our model slides, though, look quite a bit different. They're simpler. They have this feature that I like to call perfect contrast. There's no ambiguity about a blue pixel versus a green pixel. Whereas in a histology slide, there's a great deal of ambiguity about exactly which shade of purple are you talking about, and how much did that purple change from laboratory to laboratory and stain batch to stain batch. So I think I will finish there. I acknowledge my funding from NSF, EPA, and the NIH, and people, in particular Professor Glaser at IU, and numerous members of the Glaser Group, past and present. This particular project, it's kind of been my Friday afternoon project for the last year or so. And so there hasn't been a lot of collaboration on this, except for the original collaboration with Dr. Matsuura in Japan on epithelial tumor topologies. So I'll stop there, and I'll be glad to take any questions if anybody has any.
Great. Thank you very much, Jim. Floor is open for questions.
Hi, Jim. This is Yi.
Hi.
Very beautiful talk. Every snapshot you have is a snapshot in time too.
Yes.
How did you decide? Yes, all these figures, right? They are all evolving in time. Some may have arrived at steady state. How did you choose what figures to use for such comparison?
That's a great question. And in fact, that's kind of the challenges with this particular model is you see random sort very quickly, it does a lot. That's actually a lot done. And then it takes 95% of the rest of the simulation to get to the end. And
right,
I basically
right.
Yeah.
So it's a transient towards some steady state, right?
Right. And what I'm picking is just the end one. So what I did is I purposely picked a very large number of steps to try to ensure that the simulations had got to a global minima. They didn't all anyway. Many of them did, but some of them didn't. So that's the thing. You just have to make that choice of, I'm looking at my end shot. One thing I'd like to explore, and it's a little bit different of a problem, but now that I have this set of images, it would be to ask the question, if I have this image, I'm not going to predict the end image. And that'd be extremely difficult.
Right.
What I think I can do is take this image and predict the end cluster. Predict that it's going to end up, that image is going to, another 90,000 Monte Carlo steps is going to end up in this cluster. It's going to end up in this cluster. So to short circuit having to do the full simulation and do a much shorter simulation and predict forward. But yes, you do have to pick a point in time.
Okay, and just looking at your picture now, what you call as pandas or sock doll, are they stable? Right?
Yes, these down here are stable, with the exception that little balls are going to drift relative to each other. What might be unstable are ones like this one here. Given enough time, those might have fused. Same thing with this one. And you can even see that in the original simulation that very late on,
Right.
you get to that point.
Right.
And now it's this is a local, it's trapped in a local minimum
Sure.
for this parameter set. It'll get there eventually, but maybe I didn't wait long enough, and maybe that's what some of those clusters are. It's interesting to speculate about whether or not biological systems get trapped in local minima.
That was what I was just going to say. If your goal is to compare this to the physiological slides that you see, the pictures that you can have in pathology, all the images are snapshots anywhere between in the progression of the disease.
It's true. And they're 2D. Yeah.
And they're 2D cross-section of 3D images, right?
Yeah.
So does it make more sense that we actually look into the transient images rather than looking at the steady state of your simulation?
I think for cell sorting, you are looking at a system that is going to some global minimum.
Right. What I'm saying is,
and I agree that in this case, you're probably right, that in the real world, it might be going to a global minimum that's death, and you probably don't want to model it at that stage. But yes, that's an interesting point, that what this looks like, for instance, does this progress to this? And depending on where, at what point in time you actually had your histology slide taken, it looks like this, but it's going to end up being this. Well, that comes back to, can we predict forward that from this image very early in the simulation, which would be a histology slide taking at some early time point, and
right,
we predict that it's going to get to the cluster that represents much later, some point later in time. That's an interesting idea. I need to think about that some more. And I just crashed PowerPoint. PowerPoint does not like these embedded movies.
Okay, a related question. Sorry, I'm speaking too much. A related question is actually in RNA vector interpretation. So you can have single cell RNA-seq, right? And then from that, you can, like what you had done with the minimal tree or how these different clusters are connected, then you can interpolate the pseudo time. If you had any idea about any of the cluster, how they are related in terms of developmental stages, then you can infer a pseudo time. And I think it's potentially possible in your case.
Yeah.
I think so. I think it'd be interesting. It'd be interesting to use the single cell RNA-seq basically as colors on the image.
Right. Well, eventually it comes down to your clusters and how closely they are related, right?
Yep.
And if you have any idea how these two clusters should go from one to the other, then you have a pseudo time. Then you can infer the evolution.
Yes. And I'm thinking about how to actually go about doing that.
Right. So that's a good idea. This is Chang. I think, Jamie, what you can do probably instead of waiting to the last time point, you can just get snapshot along the way. And then, and those one have different patterns, and then you can connect them, I guess.
Right. Use all the transient data for your clusters too. Then you know exactly how the time will evolve.
Right. Yeah. That's a good idea.
That's an interesting idea. So, like I said earlier, is this like this solution right here? It just didn't have long enough, and it would eventually end up in this cluster.
Right. Then you have a direction of time.
Yeah. So I have the full set of images, or images every 1000 MCS or something like that. So I do have that. So for this image, I could stack behind it all of its precursor images, including just one early image, and see if there's a trajectory from the early image to this. And if you can make a prediction that given time, this trajectory would have continued into this.
Exactly. Yes.
That's a great idea. Thank you. Think
Mhm.
about how to actually implement it, but okay, I think about it. Thank you. Great idea. Go ahead, Lorenzo.
Yeah. Hi, Jim. Thank you very much. It was really good stuff. And I'm asking two related about the classification problem. So with the BG16, you take an error net, you remove the last layer, and then you plug in a new classification layer. Did I got it correctly?
No, no. You can do that, and actually have another project where we do. We basically plug in a new layer here and retrain the network starting with the pre-trained VG16. That's good. This is called transfer learning. You have a
That's
trained network,
where I
but
was going. Yes.
yeah, you're going to change the input, and you're going to change the input images, and that means you have to kind of recode your output. But
You
you're
can do some
leveraging.
fine-tuning, intercept in later layers. Just keep the big part that recognize pattern in the image. You have your last a few but you need up to plug in a little bit of knowledge of your particular image for the little that I know about this stuff. And otherwise, it will try to apply his knowledge about cat and dogs to your problem, and it might not be relevant eventually.
But we could take this vector of 1,000, remove it, and make a vector of 32. And then what those vector of 32 is just the cluster IDs.
Yeah, your friend.
So
Those things that you know that you like to classify.
Right. It wouldn't be generating a prediction of the output image. It would be generating a prediction of the output cluster. And that's why, and the one I was thinking about is, so instead of giving my 100K MCS image here and have 32 nodes here, what we do is you put the 10K images here, and the cluster is determined at 100K MCS. Again, we're trying to predict forward in time, saying if at 10,000 Monte Carlo steps the image looks like this, which of the cluster groups at the end is it going to look like? And that would require training. However, it's transfer learning, so it's certainly doable. I'm sure I could do it on my laptop, computationally a couple of days. It might take me 6 months to get the code to work, but once it was all set up.
The images are not huge, so normally. So if you are a 512 by 512, it shouldn't be super heavy for a decent GPU. A decent GPU on a laptop, I mean, not something. Well, my second one, just to not steal all the time. Since
and it's transfer learning. Yeah, it's transfer learning, so it shouldn't be so bad. I mean, I wouldn't want to go into my laptop and go at 1 million
from
input
scratch.
Yeah, that's not going to work. But here, oh, absolutely. You've got 700 or 1,000 input images, and you're going to have an output vector of 32, and you just want to retrain. Yeah, very doable. And you could do that either to make a fast interpreter for final results, or you could do it to make a predict forward in time with an early slice here, trying to predict
when you give example of stuff happening in time, essentially.
Yeah.
Well, the connected one was about just the classification. Again, since you have images that are not big, quite well contrasted, as you mentioned already, so you wouldn't be afraid of overfitting in this sense because it's perfect contrast. Would a random forest trained on your image that serve you well?
I think it might. You're right that our images, because they have perfect contrast and because you're not trying to pick out, where is the subtle difference significant, where is the subtle difference just totally irrelevant, like it is an H&E slide. Yeah, I think random forests would probably, and I should try the random forests. It would be easy enough to do it on this data. It's a little trickier to do it on the image data, but it probably can be done there. It certainly can be done there with Where do I want it? It's we certainly do random forest on this vector.
So there are
Remapping.
image classifier based on random forest.
Yeah.
And you essentially the game is that you transform the image in such ways that is informative. So one of the easiest one is blurring, so that it becomes some a green and red blurrish, and then this random forest they are normally
Yep.
pixel classifier, and they are very good at once you know what you want, they are very good. The only problem is that when you are afraid of overfitting, you have problems. But anyway, thank you very much. That was very interesting.
These images have cell outlines. And I considered running it without the cell outlines turned on. But I think that really changes the problem, because without cell outlines, this image, for instance, just becomes too blob, a blob and a green thing wrapped around it.
But if you use a Gaussian blur at different degrees, you lose the outlines and you keep the color.
Yep.
So if it's a problem, you lose it tweaking the system somehow.
Yeah, and
Anyway,
it's
good
unclear to
luck.
me. Yeah, it's unclear to me that you need the cell boundaries. It's clear to me that they would make a difference. It's not clear to me if it's a good difference or a bad difference.
No,
Maybe,
no, it's just a classifier. These are good. When you say, I take what you give me, I might go back to why you make the decision, but then it is what it is. And they don't
yes, that's right. It
what
doesn't tell you what it is. It's actually what
it
it's
will
seeing.
become or something. Yeah, no, it's a completely. Thank you.
Other questions? Lucas, you had a question?
Yeah, thanks for the great talk. Really enjoyed it, and I think it's nice that you bridge these ABM or CompuCell simulations with image classification. So that's a great idea. And I also like the spanning tree as a visualization method. I'm not sure if you've used it somewhere else, but I haven't seen it this way before. So that's a great idea. Yeah, like I had a few more general questions on the implementation pipelines that you used, or I think in principle you mainly considered one pipeline associated with the VGG16, right? So you first had the it's basically neural network based dimensionality reduction, I guess. And then you go into PCA, into the clustering. If you would remove the VGG or other parts of this pipeline, would it still work well? Or do you think that you already alluded to that some parts could be replaced, right?
Yeah, well, so there's kind of two responses. One, I use VG16 just because, I don't know, I picked it at random to start with. It could have been ResNet, or there are a lot of other convolutional neural networks for image classifications. Hopefully it's 6 one, half dozen of the other, though I don't know that as fact because I've not tried ResNet. You could imagine any number of other dimensionality reductions. Clearly, though, you can't just take your 150,000 pixels and feed it into PCA or into k-means. I don't think it's going to do anything of value. It'll do something
because you lose the context, right?
Yeah, there's no context in the image. K-means may start doing things like trying to, which images seem like they go up and down, which images seem like they want to go left or right. It might be able to do, this is a lot different than this, even if you don't know anything at all about the numbers, just because this has a lot of black next to non-black.
Right. And so for the image classifier, do you really need a color channel? So would it also work with grayscale, potentially, at least in this application?
Yes. And in fact, you could do this all in grayscale.
Okay.
Because in fact, like I said, these images have perfect contrast. So
right.
Conceptually, I should be able to recolor this image here to be black and white. Black is 0, green is 1, and red is 2. And it's
Okay.
grayscale, just 0, 1, 2. It doesn't even go to 0 to 255, just 0, 1, 2.
Okay, okay. I mean that's also useful, right?
Yeah.
For if you want to, yeah, and that was my last question. So in the future, then I understand you're right that you were also thinking about really doing like classifications or predictions directly using the neural net without adding an additional pipeline. So just trying to find different classes or predicting the behavior of the simulation, because then you would really need to train something, right? And then it might be easier to use.
Well, yeah, maybe. I think there's two applications. One is just given a model, given the things that I've included in the model, what can the model generate? So here we see this is what a very simple cell sorting model, which really it has cell volumes and it has, but adhesion energy is really about the only thing going on in the model. And this is the range of things that it can generate. If the model is more complex, and particularly if you start thinking about, well, what if my model has epithelial cells? Well, so now one of the parameters you sweep is the epithelial cell's basal level adhesion to the basement membrane. What happens if that adhesion gets bigger or smaller? And that's going to generate some set of outputs. This model here already has that included in the model, but normally if you're running the model and you change the parameter in your epithelial layer, lift off your basement membrane, oh wait, the model's not supposed to do that. So I'll put my parameter back where it pulls it back down where it's supposed to be. But there may be cases, disease cases, where that is an emergent property. So starting with an epithelial layer that has abnormal growth, what are the different things that you can generate with it? How many of those different things actually map to biologically observable defects, disease states, or something else? In this model we just published, our original model could do the first two and the last food. We couldn't do this one because we didn't have mucus secretion by the epithelium in the model. And it turns out the reason you get a mucus-filled cavity is because the cell has lost its ability to tell which way it's supposed to pump mucus. It's supposed to pump it out, up into the extracellular, up into the lumen, and instead it's pumping it down into the parenchyma and into the stromal cells, and so this emerges. In any case, that was my original thought. You've got a model and it reproduces normal in one disease state. How many other disease states might you be able to get?
Okay. Yeah, thanks. And I will post a link to a related image classification paper in the chat. Some of my students used that a while ago. I'm not sure, you probably already knew about it. It's called Swin Transformer, and they were able to, from scratch, so they participated in the Kaggle challenge, and they were among like a global one, and they were among the top 100 teams, and they used this new, 2 or 3 years ago, new transformer image classification technology, and just wanted to share it with you because you may find it useful.
Yeah, I'd love to see it. What kind of images were they working with? Were they some kind of
so they had also animals, so it was for some kind of, I think it was not cats and dogs, but a more general animal data set that they used to train it and classify different images of animals. And it worked pretty well. And the training times are significantly lower than for some of these convolutional architectures. And the overall performance is also a bit better. So I think it's a good trade-off.
Well, great. I'll take a look at it. Thank you for sharing it. One of the challenges in a project like this is, I'm using this pipeline, I'm using VG16. Should I use ResNet 34? Should I use one of these other existing, image reclassifiers or image vectorization routines? And the hope is that it doesn't make too much difference. There might be one that's faster and more effective, but in general, you get similar results no matter how you do it. But we don't know that's true, and it might not be the case. And
That's
so, I'd love to take a look at it.
that's the case when you get a few undergrad students to run it. Try them all.
Yep. And it's a great project. It
Yeah.
is really a great project, especially given these days with Jupyter notebooks, and you can, probably find a demo somewhere out there in the wild that gets you 80% of your code, and you can start then just focus on, well, what if I swap my image classifier? There we go.
I have a question, Jim. Thank you for this very fantastic talk. So I know some of these images are intermediate states, right? Haven't reached steady states, but have you tried like based on your classroom results to look at how the parameters are clustered? Like what set of parameters, right, leads to these patterns, kind of patterns? Yeah.
Yes, James keeps suggesting something along those lines.
That would be something ultimately important, interesting. Right. Yeah.
Yeah, I think I think they certainly do. But I think we know from the starting point that the interaction of the parameters is relatively complex. There are a lot of, it doesn't matter what this adhesion is. What matters is that the ratio of these two adhesions is such and such. And so it's certainly a nonlinear interaction.
Okay, so maybe there's a relationship between different parameters, right? Which get clustered.
And this would actually be a great place to try some of these neural networks that are actually designed to try to figure out the underlying equations.
Yeah.
Here's my five parameters. Is it linear in all five? Is it the ratio of the first and second times the third to the fourth power?
Yeah, that'll be really interesting to explore.
Yeah.
Thanks.
Yeah, you took my question away, which is inverse problem. If you give them an image, can you reconstruct at least probably because they're equilibrium images at some level, you can't reconstruct the absolute value for the parameters, but you could reconstruct the ratios of the parameters, as Jim said.
Yeah.
To some extent, the clusters that you're seeing here correspond to the things that we know about the physics.
Yeah.
Does green wet blue? Does blue wet green? Does green stay connected? Does blue stay connected? Are you above or below the Young-Laplace wetting criterion or equilibrium criterion for each pair of interfaces? And so you have full sorting, you have partial sorting, you have dispersal of one species, you have dispersal of the other species, you have dispersal of both species. And so in fact, you're basically seeing that it is in fact very nicely putting together the physics it's clustering the physics that we know. And so that's very nice. So in principle, it would not be crazy to ask it to come up with the ratios between the parameters or the relative distance. Maybe sometimes it's not the ratio, but it's the difference between two parameters, because the surface dimensions are the differences between numbers, between the energy functions.
So one way kind of, let's see what I want to do. I want to do the distribution. So the distribution of, these are the input parameters. Right? And terribly boring bar graphs, but that's because, it basically sampling, for a given set. But those are the input parameters. And then I can go,
sorry, did you use fixed values of each input parameters, 3 levels, or did you pick randomly low?
No, it did 5, 10, 15. Okay,
because it might be more interesting to actually spread the values so that it's not just always the same 3 values. I don't think in this case you're going to miss anything by doing that, but at least in principle, you could miss something by right. There could be, if you're doing with that level of granularity, there might be qualitative solutions that you miss completely.
Yes, you could fall through a hole.
Right. Because they don't have that big a parameter space. But I think it's really it's impressive how far you've gotten. The integration into the workflow is impressive to me. And I think it's something we could apply to a lot of different problems. There is this issue, as you point out, which is that you're not running it to equilibrium.
Yeah.
Therefore,
Well, yeah, you're not sure if you're at, there's no way to know if you're at equilibrium.
I mean, at some level, certain of these things never get to equilibrium. Some of them get to equilibrium, but the cell sorting slows down exponentially in time.
Yeah.
So when you get to you see those two clusters and it's an activated process, at some point the barrier between them will break, but that could take a very long time to happen. And so I think that this idea of trying to look at the dynamics and ask the question, can you predict the classification from early? That would be very interesting. Can you invert it to get the parameter sets, or at least the ratiometric or differential parameter sets would be very interesting. I wonder, are there workflows that actually could use movies? In other words, something that's essentially a generative model
Yeah.
where multiple images in sequence and ask it to predict outcomes or to classify, as opposed to giving a single image.
Yeah, there must be something like that, but I'm not familiar with it. What I was going to show here, so here's JCC value. So if I click on this, it tells you these are all the simulations, since I'm doing basically a Latin square here. If JCC is 15, I get, all the other parameters because I'm exhaustively stepping through it. But if I pick just this high value and you look at how it distributes across the clusters, it's all over the place. If you pick a cluster, it shows you how these values distribute. But this cluster is biased towards JCN being 5. But there is much less bias in the other numbers.
Right.
And you can either say that, well, that means the other numbers are not significant. And no, actually, it doesn't say that. It just says that there is no linear model that fits that. But there may be some combination of these values. That if it's this, it must be this, and you can't see this in this kind of analysis. So there are higher dimensional interactions that are, it's almost nothing. So this cluster is pretty heavy. So 15 for JCC, 5 for JCM, 5 for JNN. It gets whatever this cluster is. I don't remember what this cluster is. Anyway, yeah, it would be great to be able to do a time series, and it'd be great to see if it could start to predict what the interactions between the metrics are, or at least suggesting what they might be. I know there are codes which will just do exhaustively. You have 5 input variables, well, 5 linear, the squares of each, the cube of each, the sum of the sum squared, the ratio, The problem with those is that neural net doesn't really help. But the problem is that suddenly you have your column of 5 values turns into 1,000 columns.
Right.
And now if you do, p less than 0.5, well, you got 1,000 columns, you're going to get 50 statistically significant correlations if you don't do your statistics right.
I think, we had the talk 2 weeks ago, talked a little bit about this issue where you start with the linear model or the linear model plus the first order products, and then you ask how could you correct on top of that?
Yeah. Actually, that's a good idea.
And so in this case, ironically, this was a problem that Kevrakides wanted to work on 20 years ago, using neural networks of that day, and we couldn't think about what the ODEs should look like. Was a mistake not to do that, actually.
Mm-hmm.
But it's impressive to me that you get this clear clustering and classification. So it does suggest that actually you could ask the question. The other thing, if you try doing, it's not quite the same, but so the way, if I had an image that was not in my training set, and I brought it to the classifier, I'll show it to my network. It'll give me my 4,000 numbers. Then I'll run it through your PCA, and then you'll do the clustering, and you'll say it belongs to this universality class. It belongs to this cluster. So that's a practical way then to ask the question of what is the output of a new simulation. So I can now ask the question, did it fall in this class or not? It would be nice again to be able to do the inverse problem. The other one that would be, I guess, trying to think about, would be nice to be able to do, which would, I think, unfortunately require training, would be if you did it as a GAN, then you could give it your latent space vector and ask it to generate an image. Right? You can't run your network backwards here.
Right, right.
If you did it in a GAN context, then your latent space classifier would then also be able to go backwards to generate a real space image. And that could be pretty interesting because you could ask a question like, if I give it some set of values in that latent space classifier that were not in the training set, what kind of image did it give me back? Maybe it'll tell you something about possibilities that were missed in the training set.
So in a training mode, so we
Right.
can replace this 1,000 vector
Right.
with parameter sets. So
Right.
or by 3 adhesions of 5, 5, 5, 5, 5, 10, 5, 5, 5, 15, da 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 da.
Right.
We train, we give the output image and we train it to predict the input parameters.
Right, but then again, you'd have the inverse mapping where you were going from the vector to generate an image.
Yeah.
And that's the missing piece. You don't have the backward.
Yeah, this guy can't go backwards. He could look at, if these were not experimental, if these were, histology slides,
Right.
and we had some model that reproduces the histology biology,
Right.
we could use our model to train forward.
Right.
And then when you put a histology image that has not been trained on, it makes a prediction of, well, these are the, in the simple case, these are the adhesion values that reproduce, that would create this tissue layout.
Exactly.
Yeah.
And
But you're right, a GAN would be more elegant if you could get it to work.
but I think that there's a lot to do going forward with this.
Yeah.
I mean, let's follow this up and have a conversation about this.
Okay. I think maybe we can get an undergrad to set up a web page that basically has this Jupyter notebook in it. And somebody can just come in and say, well, here's my 50 simulation results from Computational, or, any other model. And it just online, do it as an online tool. Upload your 50 images and here we go. Okay, yeah, it'd be a good idea. It'd be a great thing for maybe an undergrad or a master's student in AI/ML that just needs, a project.
Okay, are there any other questions for Jim today? We've had a great conversation. Okay, I want to thank everyone for coming. I look forward to seeing you in 2 weeks. And thank you, Jim, for the great presentation.
See everybody later.